Hello friends, this is from Chambers, and today we will be continuing the discussion on general pharmacology, and we will be finally ending the general pharmacology in this video. So we had read about all that maintenance doses, steady state, half-life, rates of elimination, the first order kinetics, the zero order kinetics in the previous videos. So today I will be starting the discussion with uh, enzymes. So you must have read that in your biochemistry. So here I will give you the pharmacology association. So let us share the screen. So friends, if we think about last time we had said that in the pharmacodynamic processes, we are seeing the drugs, the effect of the drugs on the body. So we said, they can act either like an antacid, they can act like a, competitive or non-competitive inhibitors, they can act like osmotic diuretics by physically increasing the osmolarity of 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 that region and then pulling the water towards it, and then you get the all the fluids, edematous fluid out of your body. Now one part of that is remaining, that is enzymes. So in the pharmacodynamics, so enzymes, they can act, enzymes can be, the drug can be a competitive inhibitor of enzyme, or they can be non-competitive inhibitors. Okay, they can be either competitive or they can be non-competitive. So drugs can either inhibit the enzymes competitively, or they can inhibit non-competitively or uncompetitively. But here I will be discussing only the two, that is competitive inhibition and non-competitive. I would like to have some differentiation between the two also. So if I talk about, this was for example an enzyme. This is an enzyme. Okay, this is an enzyme. Now this enzyme has a place that is known as the substrate, the place for the substrate, isn't it? So once it comes to the substrate, this ice cream, okay, this substrate is coming towards the enzyme. It forms the ES, enzyme substrate complex, forms the enzyme substrate complex. So enzyme plus, let me make a different color, enzyme plus substrate will form the enzyme substrate complex. Easy. Now, if your drug is having the same, can you see the structure is same? Here, the structure is same, then it can bind like the substrate. The enzyme will think that my substrate is coming, but who is the substrate? Our drug. So if I make a drug which is in this very cone form, if I make a drug which is in this very cone form, so it's okay for me that I can directly attach that and then inhibit the drug. So this active site, this site is called the active site, isn't it? The place where the drug is binding, that is known as the active site. So can we see that it is a competition? Can we see it is a competition? Why, sir? Sir, there is competition. If I talk about, there is some sort of a competition, isn't it? How? Sir, if I have a drug which is in that, in that shape of the similar structure, then it will, bind at the active site, isn't it? Isn't it? So I had taught you last time the sulfonamides. Do we remember the sulfonamides? Sulfonamides with the albumin. And I told you, once you give sulfonamides, it will lead to unconjugated hyperbilirubinemia. It will lead to unconjugated hyperbilirubinemia. In this competitive type of inhibition, what is happening? We say a term that is surmountable. We say a term that is surmountable. What is surmountable? Surmountable means I can reverse. I can reverse if I have attached the drug, if I have attached the inhibitor at the active site of the enzyme. If I want to remove that drug from that active site, from this active site, I can inhibit. Okay, isn't it? I can inhibit now further binding of this by increasing the dose. So for example, I had a drug A and I had a natural substrate in the body, that is B. So this was natural for me, and it was binding to the enzyme. It was binding to the enzyme here, black one. But now I gave a drug, and this drug is coming to this active site and competitive inhibition, and will bind here. But if you want this pink one to get removed, you will increase the dose of the substrate, isn't it? You can increase the dose of the substrate, the real substrate. This is a drug. So that means it is a sort of, it's a, I can say it's surmountable. It is surmountable. Okay. So here two things were binding at the same site. There was a site competition. There was a site competition. Okay. Now there is a concept that is known as Km. Km is known as Michaelis Menten constant. Michaelis Menten constant. Michaelis Menten constant is telling us that how much amount of substrate do I need to produce the maximum velocity, but not the maximum velocity, the half of the maximal velocity. Means, again I will say, Km is telling me, what is Km telling me? Km is telling me, this constant is telling me amount of substrate which I need to produce half of the maximal velocity. Are we able to understand? So we have two terminologies, Km and Vmax. Km is telling me that how much amount of the substrate do I need to produce that velocity of the reaction, but not that half of the Vmax. So not Vmax, half of the Vmax. Are we able to understand? Some people feel it difficult. It's just simple. You have to reach Vmax, for example. You have to reach Vmax, but you reach this. This will be half of Vmax. This will be half of Vmax. So Km is telling me how much amount of things do I need? How much amount of petrol will I need to reach here? Are we able to understand? So can I say, if so, how I remember this Km? It is as simple a natural human brain can perceive is this is Km. So we used to write in school kilometers, isn't it? Kilometers. So this kilometers, how do I remember? This is kilometers. So kilometers, that means when you are, in the race, you are in the race and you all are standing at the start line. Okay, this is the start line. So what you will do? You will be in competition. That means you will have to run more kilometers than others. So you should remember, in competitive inhibition, Km will increase. Km will increase. So if they ask you what will happen to Km, you should say Km increases. Simple. Just remember it like this. If you can't go into the chemistry. Okay. So in competitive type of inhibition, there is increase in the Km. There is increase in the Km, but his Vmax unaffected. No change. No change in the Vmax. No change in the Vmax. Can we now remember the competitive? If I ask you in the competitive inhibition, what will happen to the Km value? You should say, sir, it increases. Directly, competitive, you should remember competitive is competitive, remember. Okay. Vmax, same, not affected. No change in Vmax. Easy. Now, if I have a drug now, and it is, it is having so much high bonding with the enzyme, it is making a very high bonds with the enzyme. It is making very high bonds with the enzyme. I can't replace. I can't replace it. I can't remove it. Whatever I can do, I can't remove it. Even if I increase it 1,000 times, if I increase another drug 1,000 times, I can't remove it. It is having such a strong bond. This concept is called irreversible competitive inhibition. Please remember, if a drug is very strongly bonding with the substrate, if I have a very high drug is having a highest binding capacity, so I can't replace it, I can't displace it. Even if I increase the number of substrates, if I increase the number of the substrate, I can't displace it, I can't displace it. So what will happen? That will result in an irreversible competitive inhibition. So now tell me, will the still Km rise or not? will the still Km rise or not? Because at the same time I have the competition. So I have to remember Km will increase and Vmax will decrease. I won't be reaching to that maximum velocity. Come what may, come what may. I won't be reaching to that maximal velocity. So in the competitive, if you are talking about competitive blocking or competitive antagonism or competitive inhibition, you should remember there is always, if I talk about competitive antagonism, I would be speaking there will be a competition between agonist and antagonist, because they have to bind to the same site on the enzyme. Once I increase the concentration of the agonist at that place, that can lead to the reversibility to the original reaction. So once I increase the concentration of the agonist, that can remove the blockage, that can remove the antagonism, I'm talking between agonism and antagonism, so that you get a very nice concept. According to first I gave you a biochemistry view, now I'm telling you the pharmacological view. So simple. Someone tells you what is competitive antagonism, sir. If I am increasing, sir, first thing, there is a competition between agonist and antagonist. Sorry. Now, antagonist will bind to the same site as the agonist or any natural ligand I have in the body. So now if I increase the concentration of agonist, that can prevent that antagonist to bind to this same place. Okay, so Km will be increased, Vmax will not be affected. But if I go into the non-competitive, if I go into the non-competitive antagonism, what happens in the non-competitive antagonism, or what happens in the non-competitive inhibition, it only depends which things you are comparing. Okay, so by the way, what was the example for this competitive inhibition? What was the example? The example was sulfonamides. Sulfonamides, I told you, it results in kernicterus, it results in unconjugated hyperbilirubinemia, it results in unconjugated hyperbilirubinemia. Okay, so if I talk about this non-competitive inhibition, what happens in non-competitive inhibition? I have this is the site again, this is the active site, this is the enzyme, this is the active site. Now this active site is meant for the binding of substrate was coming here. Okay, that substrate used to come here and bind. But what is happening in the non-competitive inhibition or non-competitive antagonism is that I have an extra site present here. This is having an extra site. Okay, this is having an extra site. I have an extra site on the enzyme that is known as the, can someone tell me what is the site known as? Allosteric site. This is the allosteric site. This allosteric site, what I can do, first tell me, is there any competition between substrate and the drug? Is there any competition between substrate and drug in non-competitive? Yes? No, sir, never. Sir, because we have an allosteric site and he's binding to some other place. He's binding to some other place. How there can be competition? So we should understand that there is no competition. So that's why we said non-competitive inhibitor. The simple concept we can make. Now the bind, if I have to give any blocker, I will be giving at this site, allosteric site. Once I give it in the hair, come what may, you increase the substrate concentration, you increase this concentration, you want to remove me at the allosteric site, you go, keep going on, keep increasing the, move on, substrate, increase the substrate, keep increasing the substrate. Still I will be working at this site because the site is different. You are not doing anything to me, but you are telling him you are increasing the substrates. It won't be helping you because my site is different. So in the allosteric mode of inhibition, if you are increasing the substrates, come what may, no substrates, you won't have any, you can't overcome this antagonism. You can't overcome this antagonism. Okay, so your Km will not be affected. Can we understand now? Your Km will not be affected, but your Vmax will decrease because I am at a different site. Are we able to understand? I am blocking at a different site, and my Km, my Vmax will decrease. Are we able to understand? There should be a crystal clear concept with this. This is so easy. The things have to be understood in that way only, in which way they are being written in the books. Are we able to understand, guys? Is it clear? There was another concept where the students are confused. I have found many students confused. They start then speaking anything of their own, then questioning me. So let us bring that concept also. There is a concept called reversible antagonism and irreversible antagonism. Reversible antagonism and irreversible antagonism. Sir, what is the difference? Now this is a cell. This is a cell. Now this is the receptor. This is the receptor. Okay, this is the receptor of the cell. Let me make it like a cup so that you can, because you love these shapes, isn't it? So this is a cup. Okay, the cup is made like it feels like he has paralytic poliovirus associated polio, isn't it? Vaccine associated paralytic polio, VAPP. So it's not like that. I have to make like this. Okay, so this is the receptor. Now what is happening in the receptor? I am giving a what? I am giving a drug. I am giving a drug to this cell. The drug is coming to this receptor, which will is equal to form what? Drug receptor complex. Drug receptor complex. In the reversible blockage, once I'm giving the drug, it is blocking the site, and whatever you give now the substrate. For example, I will give an example. Why to make this thing? This is my, let us take adrenaline. Adrenaline works on which receptor? Beta receptors. Okay. For you people right now, making it simple, not talking beta 1, beta 2, just keeping it simple. This is a beta receptor. Once adrenaline is binding to beta receptor, I will say adrenaline beta receptor complex. I will say adrenaline beta receptor complex. Easy. Now, if you have given, or sorry, if you have given this adrenaline binds to beta, now I give a drug that is beta blocker. I am giving a drug which is coming here. Before the adrenaline comes, I will be giving beta blocker. I am giving a blocker. This blocker is, for example, anilol family. I take propranolol. I give a beta blocker, propranolol. Now, guys, tell me. If I can remove this propranolol from the receptor, which is now right now it is propranolol beta receptor complex. Now this is propranolol beta receptor complex, which is a blocking complex, isn't it? It's a blocking complex. So if I have to remove this block of propranolol, can I say it is a reversible antagonism? Can I say it is a reversible antagonism, that if I am able to dissociate them, if I am able to, separate them, can I say it is reversible antagonism? Can I say they are binding with, if I'm saying, if I'm not telling it is happening, I'm saying if I can do. So I will say, can I say it is a non-covalent bond? Can I say it is a non-covalent bond is reversible? Can I say I should not worry in the clinical practice because I can spontaneously, I can resolve it. I don't need any, no, I don't want to have any anxiety about terminating the action of this drug, isn't it? So it's a non-covalent, no need to worry about this. But if I talk about the irreversible antagonism, if I talk about the irreversible, I can't dissociate the drug receptor complex. I can't dissociate naturally the drug receptor complex. And because, why sir? Because they are binding with covalent bonds. They are binding with covalent bonds, and I am anxious. It needs my intervention for the termination of its action. Are we able to understand, guys? Guys, is there any difficulty? So I hope you have understood this inhibition and all. So can I say it is an or this here? I forgot to tell you. Can I say this is an unsurmountable in the non-competitive antagonism? Can I say it is an unsurmountable? because I can't overcome the inhibition. So I have to, even if I increase the dose of the substrate, I can't overcome the inhibition. Okay. Let me talk about the, I have taught you the receptors already, I think so. So I should tell you the classification of receptors. Okay. I should tell you the classification of receptors. So let us now take the topic classification of receptors. So in the previous videos, I have taught the, the agonist, antagonist, it's +1, zero effect, 0 to +1, benzodiazepine receptors, that inverse agonist, beta carbolene, isn't it? And that pindolol and all that. I've talked about it, tamoxifen. So you can watch that video, okay? The part 3 or 4, I think. So this classification of receptors. Because we said drug is acting on the receptor, so we want to see what receptors can it bind. Some books may write 3, some books write 4. Okay, so we will mention everyone. So we have first type is G protein coupled receptors. G protein coupled receptors. Okay, so can someone give me the example of G protein coupled receptors? Yeah. Yeah. Second one is inotropic receptors. Second one is ionotropic receptors. Third one is GI, then E. So E should be enzymatic receptors. Then GI, Okay, E. The second I should be intracellular receptors. Okay. Now this, G protein coupled receptors, you are every day tackling this. I know you have been reading about this a lot. So the G protein coupled receptors are actually they are known as what was the other name of these receptors? They were known as metabotropic receptors. Metabotropic receptors. Or their another name was heptahelical. Heptahelical. Or they were called serpentine receptors. Serpentine receptors. Guys, what is the serpentine receptor? It's a simple thing, my dear. If I have a receptor, its transition in the cell membrane is 7 times. That's a simple thing. So 1, 2, 3, 4, 5, 6, 7. How we would understand? This is known as serpentine receptors. Okay. Okay, guys. Easy. So they have 7 transmembrane sparing, isn't it? 7 transmembrane spanning. Okay. 7 transmembrane spanning. Their segments are spanning through the transmembrane through the 7 times. That's why they are called as. Okay. Now what is happening if I have G? So how many types of G I have? Anyone, please? How many types of G we have? Please contribute. How many types? GS, GI, GQ, GS, GI, GQ. Okay, let us write. So GS, GI, GQ. Okay, remember this much first. Okay, so if I take them like this, if I tell you that GS should be S for stimulatory, easy. I should be inhibitory. What should be Q? What should be Q? Q should be queen. No, not queen. It should be what? It's actually activating phospholipase C. It is actually activating phospholipase C. Okay, so phospholipase C activation. Phospholipase C activation is done by GQ. Now this GS, what is it doing? GS is stimulating what? Is it stimulating heart rate? Is it stimulating brain cells? It is stimulating, mesenchymal cells. What is it doing? It is simply, you have to remember, once it is stimulated, it will stimulate what? It will stimulate what? It will stimulate an enzyme. The enzyme is known as adenylyl cyclase or adenyl cyclase, you can write, okay? Adenylyl cyclase. So this adenylyl cyclase is responsible for increasing the CAMP levels. It increases the CAMP levels, okay? And that CAMP is leading to the activation, no, of those protein kinases like protein kinase A, okay? So simply you have to remember it increases the CAMP. They increase the CAMP, okay, cyclic AMP, and then they are leading to the action phosphorylation of all the substrates. Okay, like we have the same beta receptors, I can take beta receptors. That's how we remember. I will be telling you the mnemonic also how to remember every receptor. M1 works by Q, M2 works by I, M3 works by Q, D1 works by S, D2 works by I, H1X by Q, H2X by S, V1X by Q, V2X by S. Okay, there is a big, very nice mnemonic I have taught them. So alpha 1 Q, alpha 2 I, beta 1 S, beta 2 S, beta 3 S, M1 Q, M2 Q, M2 I, M3 Q, D1 S, D2 I, H1 Q, H2 S, V1 Q, V2 S. This is how you remember everything. So I will be telling you that mnemonic. So many types of receptors we should understand. Once we remember that GS is increasing the CAMP, that's how you can remember very naturally that if GS is increasing the CAMP, that means beta 1 should increase the CAMP because beta 1 works by GS. If I say beta 2 works by GS, I should say beta 2 increases the CAMP. If I say D1 increases the CAMP, I should say it works by GS. If I say H2 increases the CAMP, I should say it works by S, isn't it? Guys, isn't it? Are we able to understand this concept, how we have to use our brain in the exam, how we have to solve the objective, and how we have to remember in the drugs in the brain when we are prescribing the patient, how we have to remember that is it GS, GQ, GI. So that is the main, what we call in the language, that's funda. It is the main funda of that. So, okay, let me say like this. First I should describe GS, GI, GQ. So GQ is actually activating the protein phospholipase, actually, sorry, phospholipase C. Okay, it's already written. So GQ is activating the phospholipase C. Now phospholipase C is getting activated. Okay, if I have GQ, I will stimulate the phospholipase C. Now this phospholipase C is actually converts my phospholipids, into what? PI. This was the compound which came from the phospholipid, PIP2. So this PIP2 is broken by phospholipase C, PLC I should write in short, to produce two things, IP3, DAG. Okay, diacylglycerol and inosine triphosphate, isn't it? So these are the two things which is done by phospholipase C. It's easy, I have taught you in the production of prostaglandins also. So what will finally happen, since it is phospholipase C, C is calcium. C is calcium. So what is happening once you are increasing the GQ receptors, you are increasing the calcium. Like you have the alpha receptors. Alpha 1 is acting by Q. That's why I am having the pupillary dilation. That's why I'm having the vascular smooth muscle contraction. That's why I'm increasing the intestinal contractions. That's why I'm having the bladder sphincter contractions, isn't it? That's why I can think of, if I think of alpha 2 receptors, alpha 2 is not working by calcium, so I can't give the example. If I talk of M1, and M1 is responsible for your what? Cognitive functions in the brain, isn't it? It's actually because of the M1 receptors that you are able to do cognitive functions. It is because of the M1 receptors that you are responsible, you are having that enteric nervous system having nice coordination. It is because of the M3 receptors that you are able to increase the sweat, you are able to increase the salivation, you are able to increase the gastric acid secretion, you are able to increase the lacrimation. It is because of the M3 receptors that you are having the gut paralysis. It is because of the M3 receptors that you have pupillary muscle contraction, meiosis you call it. meiosis, you have accommodation because of M3 receptors, you have insulin release from your body. Every organ should come in your brain that wherever M3 receptor is present, you can explain the things like that. It's natural. So beta receptors are increasing the camp, GQ is increasing the calcium. So by the way, yeah, so IP3. So by the way, what are these, what is camp? Which group do they come in? Camp, IP3, DAG, they come in secondary messengers. So they are the secondary messengers of the cell. But calcium is a hero. Calcium is a hero. He is second as well as third messenger. Please mention this point on your copy. So this is secondary messenger, but calcium is secondary as well as third messenger. Okay, second and third messenger. Okay. So you have read in that biochemistry the the subunits of alpha subunit, beta, gamma subunit. You know, then they separate and then they come together. So I hope I don't have to explain that. Okay, I have to here explain you the pharmacology. Okay, so let us give you a mnemonic of how it's a famous mnemonic actually, how we remember their receptors and their working. So first we write all the receptors like this: alpha 1, alpha 2, beta 1, beta 2, beta 3. Then we'll say, you don't have to do this with anyone, okay? Q, I, S, S, S. Okay? Okay, guys. So this is the famous mnemonic. Okay. So Q, I, S, S, S. Okay. Now these all are what receptors, by the way? These all are adrenergic receptors. These all work by adrenergic receptors. So next group I will be making. Next group, let us make the next group. Now you don't have to remember like them in the a separate way that Q I S S S. No, you will remembering in a one line like this. So you will see M1, M2, M3. Now you will say M1, M2, M3. So you will be kick, QIQ kick. Okay, QIQ is kick. Okay, now you come more down and then you write what. Once you have any exam, just write it like this at your time, because you have only 40 seconds to think. So remember, alpha 1, alpha 2, beta 1, beta 2, beta 3, M1, M2, M3, D1, D2. Then you go to H1, H2. I'll be telling you what are the receptors. Don't worry, keep writing on your notebooks. And V1, V2. Okay, now see, D1, D2. Now what is the mnemonic first? The mnemonic is QISS case, kick. Then we are going to C, kiss, kiss. Okay, C, kiss, kiss. Okay, so this becomes the periodic table. So alpha 1, alpha 2, beta 1, beta 2, beta 3, you have to say kiss. For a M1, M2, M3, kick. For D1, D2, H1, H2, V1, V2, C, kiss, kiss. So C, kiss, kiss. So this is this receptor works by GQ, this receptor works by GI, S, same like that. Okay, so I will be teaching you the ANS in detail. In the ANS, I will teach you which receptor, how to remember. I'll be teaching in the ANS videos very, very much in detail. Okay, so so we had another name here that was. Let us write for this, or here I should write. So I will write HAV. What was the mnemonic, by the way? HAV. Then was one M and M. So mom. Have one mom, isn't it? I have one mom. I have one mom, mother. So how I remember? Have one mom. So I remember by, these all. My mom is cute or not? Everyone's mom is cute or not? My dears, respect. Is everyone's mom cute or not? So everyone's mom is cute, isn't it? Because they are our mothers. So we say they have cute mom. Very nice, very patient, very beautiful. Okay. So we say have. So what are these have? They are actually coming through what? So cute, can you write cute in Q? Can you write cute in like this? Can you pronounce the Q like Q? So have one mom, which is cute. So have one cute mom, you can also say, isn't it? So what is Q? These are all receptors which are working through what? What? Please tell me. Q. Okay. Okay. The one, why do you put the one, sir? Because M1 and M3. See, 1, M. What is the position of the M now? Position of the M becomes third position. So that means M3. M3 is also Q. Are we able to understand how the things we have to work in the brain? I'm saying this is 1 M. So what is the position of M? This M is at the third position. So M1, M2, not not M2, M3. So M1, M3, and H1, alpha 1, V1, M1, and M3. So you have to remember like this: H1, A1, not A1 like this, alpha 1, V1, M1, M3. This is Q, GQ, all are cute. My mom is cute, GQ receptors. Are we able to understand? Now, now tell me one thing. The people who are mad, the people who are mad will be harmful for themselves. That is, inhibit themselves. The people who are mad, no, they are very much mad. So I should write too mad, too mad. So I have to remember M2, alpha 2, D2, they will inhibit the adrenaline cyclase. That is, they are inhibitory. Are we able to understand? Guys, are we able to understand? Is it clear? I will be teaching you in detail in the ANS. I'll be teaching you in detail in the ANS. Don't worry about this thing. Just giving you an orientation that how receptors are working. Okay. So M2, alpha 2, and D2, they will be working. They will be inhibiting the GI. Okay. All are 2. Or you can write like this in the mathematical way, MAD, MAD, and then put a 2 here. Okay. Easy. It's easy. So tasty pharmacology. Okay. Now see, if I talk about the inotropic receptors, if I talk about the inotropic, what was the receptor number? How many we have discussed? G protein coupled inotropic we haven't discussed. So let us talk about inotropic receptors. So let us talk about inotropic receptors. So if I talk about the inotropic receptors, what is the inotropic receptors? In this case, a drug will come at the site on the ion channel and will not say, please give me the G protein. I want to work through the G protein. No, it will come on the inotropic receptors, which are present on the your cell membrane, which has an ion channel. So if I draw the, you may be knowing this. Now see, I have an ion channel. Let us make an ion channel like this. This is an ion channel, okay? So think that it's constricted here. Think like that for the purpose of understanding. So it's constricted here. So once the ions will come, by the way, inotropic receptors are the fastest. They are your, airplane. They are the fastest receptors you can have. Okay. Like you have GABA, like you have NM, like you have NN, like you have NMDA, like you have 5-hydroxytryptamine receptors, isn't it? Glutamate works here, isn't it? Glutamate is working on NMDA receptors. So what happens that once these are coming, for example, the serotonin is coming right now here. So what will happen? It will open. Once it opens, the extracellular ions will get transmitted inside. So these are known as inotropic receptors. Are you to understand? Guys, is it clear? What's up, guys? So Third receptor, what did we wrote above? The third receptor. It's very much important to understand this topic, really. Okay. So third, what was the third receptor? What we are going into the microbiology. So this is okay. Third one is the enzymatic receptor. Let us talk about enzymatic receptor. Okay, enzymatic. So enzymatic receptor, the name is himself telling us this receptor should have two sides. This receptor should have two sides. One side will be the extracellular side, another side will be an intracellular side. Now, this side, for example, this is the cell membrane. Okay, and I am making a, for example, I make it like a cup. Let me draw it like a hand. Okay, like a hand. Okay. Now see what happens. I am binding a drug here. I am binding a drug here, and drug is coming on the receptor drug binding site. This site is called as extracellular site. Extracellular site, simple. Extracellular site, and inside is the intracellular site. Inside is the intracellular site. Okay. Now, can I say this is also the enzymatic site? Why? Enzyme is present here. This enzyme will get activated after this. Intracellular site has enzymatic activity. That is mostly a JAK-STAT pathway. Janus kinase, isn't it? Janus kinase or Janus kinase, you can say. Janus kinase or Janus, JAK-STAT pathway was that. So it is mostly a tyrosine kinase pathway. So what happens? The drug is coming on the extracellular side, and the intracellular side is the enzymatic activity. Once this enzyme is activated by the JAK-STAT pathway, you will be having the enzymatic action like you have. I can give you, not to say the name, but this is the animal. So prolactin, insulin, growth hormone. So these are the three hormones which are acting through the enzymatic receptors, JAK-STAT pathway. And even your cytokines, your growth hormone also will act through these sites. Okay. So what is happening, by the way? What are the steps there? You may, I think you may be knowing this from your biochemistry, that there is tyrosine kinase here, isn't it? Tyrosine kinase is like this. There was very nice diagrams in the biochemistry books. So that tyrosine kinase, when it came closer, it became like this. They became like this, and then they got phosphorylated. Your all tyrosines on the enzymatic part got phosphorylated. Now they got phosphorylated. Now they will be, there will be docking sites now. Not docking sites will be docking sites will be there, and you will have the activity. Okay, just here I have to discuss for the pharmacological point of view. Okay, so third is also done. What is the fourth? Fourth is the intracellular receptors. Now let us go to the intracellular receptors. So intracellular receptors, so beautiful concept. That's a beautiful concept, really. You want to see the God, see the receptor. Okay, you want to see the God, if you want to see the nature, then see. Can you see the receptor? Your life is on ions, my dears. Your life is on ions. Think that your inotropic receptor gets paralyzed. Done. Finished. Okay. So we have to understand the receptors very nicely so that we can appropriately give the drug. We can think that which receptor will I be giving. I can think of the mutational diseases. I can think of the genetic diseases having the mutation in these receptors also. Okay. So these intracellular receptors, what they are doing. Now tell me, I have to go inside the cell. Then I will be telling the cell, okay, get started. So isn't it a slow receptor? Isn't it acting through a slow pathway? I have to first cross the plasma membrane. Then I have to go, I'm telling it's intracellular. That means I have to cross the cell membrane. Then I have to reach the nuclear membrane. Then I have to reach the chromosomes. Okay. Or if even you tell me, sir, but what if this it is present in cytoplasm? Still in the cytoplasm, I have to cross I have to lipid, I have to become permeable to lipids, and then I have to, act there. Like you have, I can tell you an example. Like we have glucocorticoids, no, glucocorticoids, or you have mineralocorticoids also there, or you have your what to say, retinoic acid, or T3, T4, isn't it? or estrogen or progesterone from the reproductive physiology, or the testosterone, these all have receptors. If they have to reach the site, if they have to act on the site, once I'm secreting this hormone, when they have to act to the target site, for example, this is the secretory site, this is the target site, what will happen? It is secreting the hormones and I'm reaching the target site. Before starting its action on the target site, it has to pass through the cell membrane of this target site, because it has receptors inside the cytoplasm. You always keep drawing the receptors on the surface of the cell. It's not like that. Receptors can be enzymatic receptors, can be inotropic receptors, can be intracellular. So intracellular receptors, you will be checking what? The lipid solubility. Finally, what is the function? What is the function once it is reaching the the central part? It will start transcription. It will affect the transcription. It will affect the transcription. Okay. And do you know, there was in biochemistry, there was a family that was known as, that PPAR family. Do you know the PPARs? Peroxisome proliferation activated receptors. That was an intranuclear, isn't it? That was a nuclear receptor family. Nuclear receptor, not actually family, that was a super family. So this nuclear receptor super family, they were either present in the cytoplasm or they will present in the nucleus. Like we have vitamin D. Vitamin D has cytosolic. It has cytosolic receptor. Vitamin D for activating your, vitamin D is coming to the cell. If it has to activate that cell, it has to go into the cytoplasm and activate it. If you want the cortisol to have the action in your body, it should pass through the cell membrane and then reach the cytoplasm. If you want the action of PET, what is PET? PET is progesterone, estrogen, testosterone. If you want the effect of these hormones, they should reach the intranuclear space. They should reach the intranuclear space to have the action. If you want your vitamin A, if you want your vitamin A to have the action, it should reach the nucleus. Then only you can have the action. Guys, are we able to understand this concept? But I will tell you one fact from the Harrison. I think Harrison has said, he said progesterone and estrogen have both receptors. It has receptors both in the nucleus and cytoplasm. Okay, so progesterone and testosterone, they are having the receptors both in the nucleus and the cytoplasm. This is a short point for you. Okay, how we will understand. So I think the receptor concept is clear. The receptors are done. Let us take more 10 minutes more or 15 minutes so that we can read some curves. Okay, I want to tell you some 3 curves. That is the dose response curve, DRC, dose response curve. What we are doing in the dose response curve is we are taking the log of doses. We are taking the log dosage, and here we are taking the population. So DRC is telling me that what is the plasma concentration of the drug here, and what is the response in the population. What is the response in the population? So that means there will be that means there will be 2 grades DRC. If I am telling that I have a plasma concentration curve on the x-axis and I have a population concentration on the response on the y-axis, that means I will be having DRC, which will be of 2 types. That is, I will be having one which is a quantal DRC and one will be a graded DRC. One will be a quantal DRC, one will be a graded DRC, isn't it? Quantal, graded. So what happens in the quantal graded DRC is, once I increase, let me talk about the quantal DRC first. See, what is quantal DRC? Quantal DRC is I am having a sensitivity variation. What? Once I increase a dose of a drug, like I have one individual living in California, second group of individuals I have in Washington. So I have seen that there is a difference. Once I am increasing the dosages in those population groups, I am having the variation in the sensitivity of the response. Okay, okay. So do we know that we have a concept in pharmacology that is all or none phenomenon? Do we? I can give you a simple concept. I am vomiting. I am vomiting, and you give me what? Ondansetron. You are giving me ondansetron. That is an anti-emetic drug. You are giving me an ondansetron. So two things will happen. One is either my vomiting will stop or my vomiting will not stop. This is all or none principle. Either my vomiting will stop. It's not that I will have little vomiting, then I will get stable. No, it's all or none. That I will be stopping the vomiting or I will not be stopping the vomiting. That will be done by the antiemetic. Okay. So if this thing is happening, if all or none phenomenon is happening, that I will be, that also I can calculate on this DRC curve. Okay. Easy. Now response, my response. I have to see the DRC, dose response curve. Dose is I am taking the dose, response is response. Okay. Now, what is the use of this DRC? It is telling me two quantities, even three quantities, I can say. It is telling me the therapeutic index. It is telling me the ED50, that is the median effective dose. And it is telling me the LD50. This is the important concept. He is telling me quantal DRC is telling me the therapeutic index. It is telling me the ED50. It is telling me the LD50. Now, mean effective dose. What is the mean effective dose? Let us give one statement and then we can remember it. Okay. Let me talk about the median effective dose first. So we write in short ED50. What is the meaning of the ED50? You will understand right now. See, I gave a dose and I knew that it will stop the vomiting. I knew it will stop the vomiting. So the response which I was desiring, that response came in only the 50% of the population. Are we able to understand? So if I am giving a dose, for example, this is a curve. Let us make a curve like this. And what happens? Where will be the ED50? ED50 will be at this point. So this is the population percentage, 10%, 20%, 30%, 40%, 50%, 60%, 70%, 80%. So in the 1, 2, 3, 4, 5, in the 50%, 50% population showed me the response, isn't it? So if I give, now this gave me the response at this dosage. So this is the dose 1 mg, 2 mg, 3 mg, 4 mg, 5 mg. So at 4 mg, 50% people are responding. At 4 mg, 50% people have responded. Now see the comparative. Now we compare it. I saw that I have another curve and that curve started with hair. Okay. Now see, the definition of median effective dose is that what is the dose that will produce the desired response in half of my population, that is 50% of those. That means if I have 100 people, 50 people should be having the response. 50 people should be giving me the response. So now see, what is the response right now? This is the population. So can you see the concentration? Where did it reach? So I draw graph A and graph B. So this was at 4 mg and for instance, I take 10 mg. Now tell me, which is very nice dose? Is A a very nice dose or B? Is A a very nice dose or dose B? A is a very nice dose, very nice, because you are having the effect at 4 mg. 4 mg, the people are responding to you. But in the B, 10 mg you have to give. You have to give such a big dose. So can I say if I have more ED50, I have lower potency? Can I say if I have more ED50, what is more ED50? That is I have to give more dosage so that I can increase the response up to 50%. That means the drug is not potent. What is potency? Potency means the amount of drug which I need to produce a response. That means if a drug is giving me a response at a lower dose, I mean potency is very nice. Isn't it? If I have a drug which is having a nice potency, that should be used only in a small dose so that I can get a potency. But if I am telling you the ED50 is higher, that means the drug ED50 is higher. That means I have to give a very high dose to get the 50% effect. At least my 50% of the population is responding. So with that high dose, then only my 50% will be responding. 50% of the people will get the response. Are we getting the concept, guys? Yeah, okay, done. So see, now I talk about the lethal dose. So if I talk about the median lethal dose, so I should write what? If I decrease the, if I have less ED50, I am more potent, happy, more potent, done. Now I am telling you median lethal dose, median lethal dose, that is LD50, that is LD50. So what is median lethal dose? Median lethal dose is the dose that will result in the, that will kill 50% of the animals when they are getting the drug. Means if I give, if a drug is having more LD50, can I say it is safe? I want true or false. If someone is having an LD50 more, can I say it is safe? True or false? It is true. It is true, because the thing lies in the perception, how you perceive things, how you perceive things. Now see the perception. What is the perception you have to take? LD50, what is LD50, my dears? LD50 is the dose that will kill 50% of these animals. Now, if I am increasing the LD50 of the drug, that means I have to increase the dose to a much higher level. That means I have to increase the dose of a very high level. Then only I can kill the 50% people. Then only I can kill the 50% of those animals, isn't it? So if I have a very high LD50, that means even if I am taking 50 mg, I'm not able to kill 50%. If this drug was killing at 100 mg, for example, the animals, if this 100 mg killed 50 animals, at least 50 died because of 100 mg. So even if my dosage goes to 50 mg, I'm not able to reach the killing effect. So I'm safe because my LD50 is higher, because the dose that will kill the 50% of the animals is higher. So ED50 is higher means I have to reach that level so that I can kill 50%. So isn't it a safe drug? Isn't it a safe drug? Are we able to understand? Okay. So if someone is telling you the lethal dose 50 is higher, if it is more, I should say I'll be happy, safe. Why? Sir, lethal dose, to reach that dose which will kill the 50% is higher. That means I will take more doses. I need a very high dose to kill something. I need a very high dose. That is the mean lethal dose. Now there is a concept that is therapeutic index. The therapeutic index is basically the ratio of LD50 by ED50. Now what is happening that I am taking as a, they were the chemists. So what they did, pharmacologists, they made a window that is known as the therapeutic index. They said if I divide LD50 by ED50, if I divide LD50 by ED50, first thing, what is therapeutic index, by the way? Therapeutic index is telling me how much safe a drug is. How much safe is the drug? That is the therapeutic index. So it tells me about the safety of the drug. It tells me about the safety of the drug. That means if I have very high therapeutic index, that means I am safer. If I have a very high therapeutic index, that means I am safe. My drug is safe. Okay. So it's the range actually. So if I am giving a big range, if I am giving a big range of LD50 and ED50, I can say I am still in the safe zone. I am in the safe zone. Even if I increase the dosage of something, I am still in the safe zone, isn't it? Guys, isn't it? So it's a range actually. So even if I'm varying in this broad, now I am telling you this is an A and this is a drug B. And who is having the narrow? Who is having the narrow therapeutic index? Who is having the narrow therapeutic index? Who is having the narrow? B is having the narrow. So should I be worried about this if I increase the little bit of dose? Should I be worried? Okay, very nice answer. So that means I should, as a doctor, I should remember that which drugs have a narrow therapeutic index and which drugs have a higher broad therapeutic index. So I should, broad is okay, broad is okay, but lower therapeutic index, lower therapeutic index is a concern. Lower therapeutic index is a concern. I have to think about the lower therapeutic index. So which drugs are there for the lower therapeutic index? So can we see therapeutic index was is equal to, can, if we have the perception therapeutic index, we were taking Ti is equal to, yeah, toxic dose. Can we write the lethal dose as toxic dose Td for our understanding? Can we write the lethal dose as toxic dose for the mnemonic purposes? This is how I remember. So therapeutic index Td divided by Ed50. Now if you want to remember the drugs will be first let me write the drugs. Theophylline, I can remember the mnemonic. Theophylline, digoxin, warfarin, okay, and anti-epileptics and lithium. So warning, these drugs are lethal. So warning, these drugs are lethal. Okay, these drugs are lethal. So you have to remember these drugs. Please, you have to remember because they have a very narrow therapeutic index, and while changing the dosages of these drugs, you have to think a lot. Okay, guys. So they have a narrow therapeutic index, and you should remember them. There were many other mnemonics in the market. If I go into that Tripathi was writing so many drugs which I was having. Like it was said about the digoxin, anti-arithmetics, TCA, tricyclic antidepressants, methotrexate, or you have theophylline. So there was a mnemonic that Chief Minister Lat, okay, antiarrhythmics, theophylline, aminoglycosides, methotrexate. So here you can remember at least these drugs for your exam point of view. Okay. So they will ask you a value in the exam. They will ask you which drugs, what is the value of the therapeutic index when we call a drug a safe drug if its therapeutic index is greater than 2. If the therapeutic index of the LD50 by ED50 is greater than 2, we say the drug is safe. If it is less than 2, we say the drug is narrow, it's unsafe. How will you understand? Guys, what's up? Is it clear? Okay. So I hope you have enjoyed this lecture. And if you have not subscribed to my channel, please subscribe. I will be uploading all the videos of MBBS, all the 19 subjects. And we will be continuing this. Again, we were not able to finish today. We will be taking the next, in the next lecture, I will be teaching you the finishing. We will try to finish the all the clinical trials, how a drug is in how many phases it can go. Like in the phase 1 it goes into the healthy volunteers are used. In the phase 2 some patients are used. In the phase 3 we take some 5,000 patients. Like that we will be talking about the microdosing studies also, how a drug is how the clinical trials are done, and how a drug is then, how a drug comes in the market. So thank you very much, and God bless you. Enjoy MBBS.